For numerous years, extensive research in the field of pancreatic development has primarily concentrated on understanding the embryonic specification and differentiation of the various islet cells and the intricate regulatory mechanisms governing these processes. Nevertheless, it is crucial to recognize that postnatal development of the endocrine pancreas is an equally intricate phenomenon. This phase encompasses the maturation of islet cells and a remarkable increase in their numbers. A pivotal contribution to our comprehension of beta cell expansion and the functional immaturity of fetal or neonatal beta cells was made by Klaus Hellestrom and his research team. In this video, we aim to delve into some fundamental concepts derived from this early research, specifically concerning the functionality, quantity, and turnover of beta cells. It's crucial to keep in mind that beta cells undergo significant changes in their insulin secretion capacity and proliferation potential throughout their lifespan. Newly formed beta cells exhibit an immature secretory function and a high rate of proliferation. As time progresses, these cells experience an improvement in their secretory capabilities, especially in response to glucose-stimulated insulin secretion, while their capacity to replicate diminishes. Eventually, beta cells may reach a state of senescence, characterized by a decrease in secretory function, loss of replication ability, and increased vulnerability to cell death. Importantly, the age of beta cells is not solely determined by the individual's age. The gradual turnover of beta cells leads to the coexistence of beta cells of different ages within an individual. Consequently, both young and older individuals may harbor both newly formed and aged beta cells, with the key distinction being that older individuals are likely to have a higher proportion of older cells. This variation in the distribution of new and old cells can significantly influence overall insulin secretion. This complexity is further compounded by the existence of two distinct mechanisms for generating new beta cells. Despite some historical debate, it is now widely accepted that neogenesis, the process of forming new islets from pancreatic duct cells, continues throughout an individual's life. However, these newly generated cells must undergo a maturation process. In addition to neogenesis, new beta cells can also be created through the duplication of existing beta cells, with the maturity of these new cells likely dependent on the maturity of the parent cell. Consequently, it can be inferred that when immature beta cells divide, their offspring will also be immature, whereas the offspring of older beta cells are likely to be mature, although this aspect has not been extensively studied. Immature beta cells exhibit distinct characteristics in their insulin secretion profile. They display a weak response to glucose-stimulated insulin secretion, GSIS, but show relatively better reactivity to nutrients like amino acids, 1. Notably, immature beta cells not only respond inadequately to elevated glucose levels but also struggle to efficiently cease insulin secretion when glucose levels decrease, 6. The latter is a crucial trait of mature beta cells, providing protection against hypoglycemia. The maturation of immature beta cells can be promoted by exposure to glucose itself. For instance, research by Frankel and Hellestrom, 7, demonstrated that fetal rat islets, cultured with 11 glucose for just 8 days, attained near adult levels of secretory responses to a glucose challenge of 16.7, compared to controls of 2.8 millimoles. Additionally, our studies indicated that islets from 2-day-old rats, treated for 4 days with adenovirus expressing the transcription factor MOFA or T3, approached the secretion levels of adult cells. However, research by Bliss and Sharp observed that glucose-responsive insulin secretion became evident by day 14 after birth, yet full development of glucose responsiveness, seen in adults, had not been achieved. This delay in attaining full capability in a living organism can be attributed to the late generation of a substantial number of beta cells in the first postnatal month, with their secretory immaturity masking the robust secretion from a more mature cell population. To explain the molecular basis of this immaturity, numerous studies have identified individual genes with low expression believed to be involved in the maturation process. To comprehensively examine a larger set of genes, we conducted a microarray analysis of laser-captured beta cells from rats, comparing them to adult beta cells. Our analysis revealed differential expression of many genes, including several integral components of specialized NAD mitochondrial shuttles unique to beta cells. Some of these genes, such as pyruvate carboxylase, maleic enzyme 1, maleic dehydrogenase, glutamate oxaloacetate transaminase, and glycerol phosphate dehydrogenase 2, exhibited low expression initially and only reached adult levels of mRNA later. This suggests that essential mitochondrial shuttles, particularly the glycerol phosphate, malate aspartate, pyruvate citrate, and pyruvate malate shuttles, remain underdeveloped in neonatal beta cells. Furthermore, key beta cell genes like insulin, GLUT2, PCSK1, and GLP1 are displayed low expression initially and reached adult levels much later. An investigation has revealed that pharmacological stimulation can influence ductile cells derived from the human pancreas to regenerate beta-like cells that release insulin, offering a ray of hope to individuals managing diabetes. The preliminary research, led by Sophia Nina Marika at the Baker Heart and Diabetes Institute and recently reported in the Clinical Epigenetics Journal, illustrates how medications can affect pancreatic progenitor cells, reactivating them into cells that produce insulin akin to beta cells by inhibiting the EZH2 enzyme. In cases of type 1 diabetes, damage to beta cells within the pancreas results in minimal or no insulin production, leading to elevated glucose levels in the bloodstream instead of its utilization by cells. This elevated glucose in the bloodstream is referred to as hyperglycemia, with the body being incapable of using the glucose for energy. Researchers have invested significant time and effort exploring alternative treatments, such as cell replacement therapy and pancreas transplantation. However, the scarcity of donor organs has constrained the viability of the latter. This new approach investigates the regenerative potential of pancreatic cells by stimulating ductile stem cells with small molecule inhibitors, offering an alternative approach to restoring insulin production for those living with diabetes. The drugs employed in this study include a synthetically engineered drug primarily used in cancer therapy, notably effective in cases of EZH2 overexpression seen in cancers, and a naturally derived drug that has undergone testing as an inflammatory condition treatment. Sophia Nina Marika, a PhD student at Monash University working at the Baker Institute under the guidance of Professor Sam L. Oster's Human Epigenetics Program, suggests that the reprogrammed cells exhibit the capability to generate insulin, and perform essential functions, including insulin secretion in response to glucose stimulation. Professor L. Osto underscores the significance of this groundbreaking epigenetic research led by emerging scientists. He emphasizes that it may have substantial implications for improving the quality of life for individuals dealing with diabetes in the years ahead. Although further investigations are required to delve into the mechanisms and identify specific cell targets in order to enhance methods for alleviating the burden of insulin-dependent diabetes, these findings open up an exciting avenue for beta cell regeneration as a viable substitute for islet transplantation for individuals living with diabetes, bridging a crucial therapeutic gap.